Okay, so in the last video we talked a lot about RNA polymerase itself, the enzyme, the molecular machine that's responsible for catalyzing RNA synthesis from a DNA template. We talked about some important biochemical features of this enzyme. So it does not require a primer to initiate synthesis. It doesn't require a separate helicase activity, and it does't have a separate 5' to 3' exonuclease activity. So this makes it distinct from DNA polymerase in a number of ways. I did mention that it had this backtracking activity that does allow it to do some sort of proofreading using the same active site it uses for polymerization, but that's very distinct from DNA proofreading mechanism. And of course, the product of RNA polymerase is a single-stranded RNA copy of the coding strand. So whatever strand we want to make a copy of here, we have an RNA duplicate of it, and that copy is made using the template strand as our template to form the Watson-Crick-Franklin base pairing interactions there. Okay, so as I mentioned in the last video, the fact that RNA polymerase doesn't need a primer raises an interesting question, which is we know it needs to know where to start doing transcription and which strand. It has to somehow know which strand it should use as a template. And so, how is that actually specified? Why start here at some region and not other places? Why doesn't RNA polymerase just transcribe everywhere in the genome? And so in this video, we're going to talk a little bit about the architecture of the units of the genome that are transcribable and what are called the promoter and terminator sequences that can specify where RNA polymerase should start and where it should stop. And so, to think about this again conceptually for a minute, to transcribe any specific gene or any specific region of the genome, what we would really expect is that there should be a start here and a stop here signal near the ends of the thing that we want to transcribe in order to direct RNA polymerase. So if there was some gene here in purple, and let's say the coding strand is here where it says gene, and this is the reverse complement down here, ideally in the genome there should be some signals up here that say this is where I should start, and there should be some signals down here after the gene that say let's stop transcribing. That way when we want to transcribe this gene, we always make the full thing and we make it in sort of the same way every time. In addition, we also need to know which strand to use as a template. So we don't want to somehow transcribe the wrong strand by mistake, right? And so it turns out these start signals, which are what we're going to talk about first, are what are called promoter sequences. And so the gene promoter is a DNA sequence which specifies where RNA polymerase should initiate transcription. So again, if we want to transcribe here and make this kind of yellow-orange thing as our gene, there's going to be a promoter sequence here, which is the going to direct RNA polymerase where to start. And more generally, there can often be other types of DNA sequences that are near the promoter that can also affect whether or not genes are transcribed. But the core unit that specifies whether or not an RNA polymerase can transcribe is going to be this promoter itself. Okay, and at this promoter, that is going to be the site here at which we end up forming our transcription bubble here and transcribing our RNA DNA hybrid. And so what are these promoter sequences? Well, they are specific sequences at defined distances from the intended transcriptional start site. And so there are going to be elements of sequence, for example, that are 35 nucleotides upstream of the start site, or 10 nucleotides upstream of the start site. And this specific positioning is basically going to kind of provide a landing strip, or basically a set of spaced sequences that the RNA polymerase can bind to. And when it binds to those sequences, it's going to position the overall polymerase in such a way that the active site is basically poised here to initiate at the specific location of interest, as well as automatically, as a consequence, use the correct strand as a substrate. Okay, so by putting these sequences at specific distances, when polymerase engages, it's positioned to transcribe right from the start here at the right location. And once the promoter has been recognized transcription can initiate. So when it binds these sequences, RNA polymerase can then open up the DNA that's in those regions to start to form the bubble. And once that opens up, as we mentioned before, there's no requirement for a primer in RNA polymerase. And so once we open up this bubble, RNA polymerase can begin synthesizing RNA and making this RNA strand. And once it gets going, basically, then the polymerase can, do the elongation activity that we've discussed before. So basically, can unwind the DNA in front of it and continue making RNA as it goes. Okay. Now, I have to say that there are, that's conceptually what happens, and that's true for both prokaryotes and eukaryotes. It's important to note that there are pretty significant differences in the definition and organization of promoter sequences between prokaryotes, so like bacteria and things like that, and eukaryotes like ourselves. And a general theme you'll learn in this course is that the prokaryotic version is always easier to understand, and in the eukaryotic version, the ideas are the same, but the execution is much more complex. And that's exactly what we're going to see here with promoter stuff. So in prokaryotes, your RNA polymerase is composed of many subunits, and one of those subunits is called a sigma subunit. And the sigma subunit is what actually is responsible for recognizing those specific DNA sequences that are at the -35 and -10 position on the DNA. So sigma factor here interacts with the DNA and it reads them out, and that's what provides the specificity for where to initiate. In fact, beyond that recognition, sigma is not needed. The other subunits of the polymerase can perform all the necessary catalysis. And so this sigma factor acts as sort of like a module to direct RNA polymerase where to go. And what's kind of cool about those sigma factors is that the sigma factor itself can kind of be swapped out for other sigma factors. So if the cell needs to dramatically change what types of genes can or can't be transcribed, a really dramatic way to do that is to change what your sigma factor is to direct it towards other types of sequence elements to sort of globally affect what genes will be transcribed. And so each sigma factor that a bacterium or prokaryote has directs the RNAP, the RNA polymerase machinery, to different transcription initiation sites. But in general, you have this nice clean thing where it's sigma factors specifying where to go, and then the rest of the RNA polymerase is the same. Now, in eukaryotes, things are more complicated. Eukaryotes actually have 3 different RNA polymerases, Pol1, Pol2, and Pol3, and each one of them has its own completely unique initiation specificity requirements. So different types of transcription factors or helper proteins are needed to get the polymerases to recognize different promoters. And on top of it all, the transcriptional regulation can be regulated by other pieces of DNA, sometimes that are very far from the transcription site. And so it's very complicated, but conceptually, and the main thing that you'll need to know about it for this series of lectures is that at the end of the day, it's a very similar concept in that you're going to have helper proteins that specify which locations, which binding sites in the genome polymerase should sit down on, and that's going to facilitate transcription from taking place. So that's the main thing I think you should know for eukaryotes for now. And regardless of whether you're prokaryote or eukaryote, once you open up the DNA, once you've bound to your promoter and you melt that double-stranded DNA segment to make this bubble, then the polymerase is going to enter this elongation mode in which the DNA template strand serves as the source on which to synthesize your RNA. And it should be obvious why only one of these strands, even so if we made this thing, you might say, how does it know which strand is which? Well, there's only one strand that's in the orientation that would be compatible with the RNA transcript being synthesized in the 5' to 3' direction, right? And that is the bottom strand here that goes in the 3' to 5' direction. So simply by opening this up, you've already imposed by the directionality here of the polymerase which way you're going to get going here. That's basically going to ensure that the DNA template strand is used to make a copy of the coding strand, okay? So that's all kind of hardwired into the promoter. The polymerase is placed in this direction, it's bound to a particular place, the bubble is formed, and then the geometry is such that it really only has one way to go. But, you know, I mentioned this fact about knowing which strand is which because there's really no rule that says you can't have two genes that are encoded on opposite strands. In that case, what we would just need is we would need a different promoter upstream of one gene positioned on so that the polymerase will extend this way, and then on the other strand we would flip that promoter sequence around and now we'd have one driving this strand. So formally you can have such a structure here in your genome, and sometimes actually you will see what is called convergent transcription like this in the genome. But it's important to remember that although we can draw it like this, it's not physically possible to have two RNA polymerases simultaneously transcribing each strand of a given stretch of DNA. And the reason, of course, to see why is that if we go back and we look at that structure of RNA polymerase and the transcription bubble that forms, the RNA polymerase actually holds on to both the template and coding strands in the complex, right? So the coding strand, so in the bubble, the entire bubble is contained within the RNA polymerase, okay? And so it's not possible for two polymerases to simultaneously be holding on to the same bubble. And so if you have this type of convergent transcription happening here where you've got two genes and the polymerases are coming at each other, eventually there's going to be some sort of they're going to block each other's ability to continue forward. And so when this happens, either the polymerases can collide and transcription for both can be aborted, or in some cases one polymerase can knock the other one off. But exactly what happens in these cases isn't important. It's just more important to recognize that by holding on to both strands at the same time, you can't really transcribe both strands simultaneously. And that, of course, is something that's different from what we learned about with DNA polymerase, where, in fact, both of the two DNA strands are synthesized coincident during genome replication. You have your leading and lagging strands being made at the same time. So again, an important distinction there to think about. Okay, so to sum up what we learned about promoters and transcription initiation, so getting the process started, we learned that in order to coherently transcribe a gene, RNA polymerase needs to know where to start transcription. There's no primer being put down that tells it where to begin. It has to know some other way, and its promoters sequences, specific DNA sequences that position RNA polymerase, that provide the initiation signals to say start here and on this strand. And once this process begins, the template strand is what's used to make a copy of the coding strand. And because the template and coding strand are both simultaneously held by the polymerase, only one strand of DNA can be actively transcribed at a given time, okay? And that's distinct from polymerase, where you can make copies of both strands. And I do see the typo here. I'll fix it in the printout of the slides. And so now we know where to start. So that's half of what we talked about before. But how do we know when to stop? So polymerase lands on a promoter, starts transcribing DNA, transcribing RNA. It's zipping along. What tells it to stop transcribing? What's going to prevent it from transcribing the entire genome? And that's what we're going to learn about in the next video on transcription termination. So we'll see you then.